Hi everybody, we are back and ready to continue our discussion of genetics in chapter 15, begins on page 294 in your text and page 91 in the course outline. And in this chapter, I do want to back up a minute to chapter 13 and look at a couple of terms that are going to be useful in this chapter. So, one of the things we talked about are somatic cells versus germ cells. And if you remember, somatic cells are body cells that go through mitosis, whereas germ cells are also diploid cells, just like somatic cells, only those were the specialized cells in the male and female gonads that go through meiosis. So most of our cells, again, are somatic cells. And those would be then any cell other than a gamete. And we have 23 pairs of chromosomes for a total of 46. And remember, you inherit half of those from your mother and half from your father. And they can pair up in what are known as homologous pairs. And those homologous chromosomes, homo meaning the same, are coding for the same traits, not necessarily the same forms of the traits, but the same traits. Like it might be a gene for eye color, but they may be donating different forms of that trait. So chromosomes in a homologous pair are the same length and shape and carry genes controlling the same inherited characteristics. So we can actually look at our 23 pairs of chromosomes in something known as a karyotype. So that's an ordered display of the pairs of chromosomes from a cell. And in order to do a karyotype, you need to have cells that are actively dividing. And there's several different reasons why you might want to do a karyotype. For example, one of the most common reasons to do a karyotype is to determine if a developing fetus is having some kind of chromosomal problem. And originally this was done via what is called an amniocentesis, where they took this long needle and put it all the way through the pregnant woman's belly into the amniotic sac and took some of the fetal cells that way. Now, this is really invasive, and so a new technique, a better technique, which is more often done, is what is known as a chorionic villi sampling. And here they can use a suction tube inserted through the cervix and get some of the placental cells, which also contain the fetal cells. So much less invasive that way. And then what they do is they take these cells that are actively dividing and they look at the cells under the microscope trying to find those that are in metaphase, because not only are they condensed and ready to go, but they are still double-stranded, and then they take a picture of those. Now this was taken using fluorescent microscopy, so you can see these different little color-coded areas are different genes. So this is a pair of homologous duplicated chromosomes that you see here. You can't really see individual chromatids here like you can here, but these again are usually done at metaphase chromosomes. And here you notice that we have 22 pairs of autosomes. Remember, autosomes are the same in male versus female when there's disorders carried on those chromosomes. And then pair number 23, and that's what we want to look at today and look at what happens when there's a faulty allele here, most specifically on this X chromosome. So this is showing you that you've got one X and one Y. So what gender is this individual? Anybody, the way that this works is in the female, these pair number 23 are the same size, same shape, and they are, since they look like they're double-stranded, they look like X's, we call it XX. In the male, there's only one that looks like that. The other is much, much smaller, and so it looks almost like a Y, so we call that an XY. So another thing you can do with a karyotype is actually determine without a doubt the gender of the baby. You can also look at all these chromosomes and see if there's any abnormalities. So you might want to look and see if there's an extra pair. So there's some disorders where instead of 2 in a pair, you have not an extra pair, but an extra chromosome. So you might have 3 in a pair instead of 2. So that individual, instead of having 46 chromosomes, would actually have 47. Or you might be missing one, so you might only have 45. Or you might have an extra copy of one of the genes, so it's a little longer, or missing one and it's a little shorter. So there's lots of things you can tell looking at a karyotype. And in fact, you have an online lab report due that will take you through an activity where you actually look at chromosomes, pair them up into their homologous pairs, and notice they're going from the largest to the smallest, same size, same shape, and then you can determine whether there's any chromosomal abnormalities. So that's what I'm going to talk to you about in this chapter. So we're going to focus also on those sex chromosomes. Again, that's pair number 23. So all the other pairs, we have 22 pairs of autosomes, and then we have one pair of sex chromosomes. And that determines the sex or the gender of the individual. And they are either going to be called an X or a Y, depending. So in the female, again, she has a homologous pair of X's, so XX indicates a female, and males only have the one X and the other we call a Y. Now, there's not a lot of important genes on the Y, but one important gene that is there is what we call the TDF gene, testis determining factor. And that is on the Y chromosome, and it codes for a protein that directs the development of male anatomical features. So the theory is that a fetus would develop into a female unless they have a TDF gene, and that then directs the male genitalia. The remaining 22 pairs of chromosomes, again, are called autosomes. So the females have two X's, the male has an X and a Y. Each ovum or egg contains an X, only X's, because that's all the female has, whereas the sperm may contain either an X or a Y. So my question is, who determines the sex of the baby, mom or dad? Right? It has to be dad, because if he donates an X, mom only has an X to donate. Remember, these separate out into the gametes. So if she donates an X and he donates an X, it's a girl. If she donates an X and he donates a Y, it's a boy. So back in the day when those kings would behead their wives for not giving them the right gender for a baby, they should have really been getting on the guillotine themselves, because it was their fault, not hers. This is the actual micrograph taken of chromosomes that have duplicated. You can see, again, it looks like an X here, and then the Y in the male is much smaller. And again, not many important genes there, the main one being that TDF gene. So when we look at how X-linked inheritance works on that X chromosome, we're going to see it's going to be different in the male versus female, because these X-linked problems are usually recessive, and those are the ones we're going to talk about, which means a female would have to have two faulty X's in order for her to get the disorder, whereas a male, since he only has the one X, he only needs one copy. So often you see these X-linked genetic disorders are only seen in males. It's much, much more difficult for a female because she needs two copies. A male just needs the one. Genes on the X chromosome then are called X-linked genes. So what's the difference between X-linked genes and sex-linked genes? So it says here a gene that is located on either sex chromosome is called a sex-linked gene. But again, we're focusing specifically on the X-linked genes. X chromosomes have genes for many characteristics unrelated to sex, whereas the Y chromosome mainly encodes genes related to sex determination, as I mentioned, the TDF gene. So X-linked genes follow specific patterns of inheritance. For an X, or for a recessive X-linked trait to be expressed, remember the female needs two copies, that is, she's homozygous, and a male only needs one, and there's a word for that, and that is called hemizygous. He only has one copy of that. Why are X-linked recessive disorders much more common in males and females? Again, they only need that one copy. So who does the male get his faulty X from, mom or dad? Well, let me ask you this. Where did the male get his Y? He had to get it from dad, right? Mom doesn't have any Ys. So what we see in these situations is the faulty X, mom has it as a carrier, doesn't know she has it, and she passes it on to her boys, which then show up with the disorder. So some disorders that are caused by recessive alleles on the X chromosome include color blindness, is mostly X-linked. There are different types of color blindness, but again, you don't often see women who are colorblind. muscular dystrophy, and hemophilia. Hemophilia was a big one that went around the royal families because, and again, showing up in the boys, by the way, that's inability for the blood to clot. It was showing up because it's a recessive disorder, and just like all recessive disorders, when close family members reproduce, then these recessive disorders tend to show up. So here's a clip on these X-linked traits.
To fly a rocket ship, you need to be an optimist. No astronaut.
While observing fruit flies, Morgan discovered a male that had white eyes instead of the typical red ones. He mated the white-eyed male with a homozygous red-eyed female. The entire F1 generation, whether male or female, had the dominant red eyes, just as Mendel would have predicted. However, the F2 generation held a surprise. Just as Mendel would have predicted, 3/4 of the offspring had red eyes, and 1/4 had white eyes. But there was a catch. Only males had white eyes. 1/2 of the males had red eyes, and the other half white eyes. If sex and eye color were being inherited independently of one another, as Mendel would have predicted, then 1/2 the white-eyed individuals should be female. Obviously, eye color was somehow linked to sex. In order to explain his results, Morgan hypothesized that while there is a gene for eye color located on the X chromosome, there is no corresponding gene on the Y chromosome. Thus, females, which receive 2 X chromosomes, also receive 2 genes for eye color, while males, which receive 1 X and 1 Y chromosome, receive only 1 gene for eye color. Looking at Punnett squares based on Morgan's hypothesis helps explain the results of his experiment. Morgan started with a white-eyed male that produced only 2 types of sperm. Sperm containing the X chromosome and along with it the alleles for white eyes, and Y chromosome sperm that contained no alleles for eye color. The female fruit fly, being homozygous for red eyes, produced only eggs carrying the alleles for red eyes. In the F1 generation, the female offspring are created when the X chromosome white eye sperm unite with the X chromosome red eye eggs. Because all the females inherit 1 white eye gene and 1 red eye gene, they are all heterozygous for eye color. The F1 generation males are created when Y chromosome no eye color gene sperm unite with X chromosome red eye eggs. Thus, all males have red eyes as a result of the single eye color gene they receive via the X chromosome of their female parent. Crossing the males and females of the F1 generation produces an F2 generation in which 1/2 of the females are homozygous for red eyes, inheriting 1 red eye gene from their mother's X chromosome and the other from their father's X chromosome, and 1/2 are heterozygous, inheriting the white eye gene from their mother and the red eye gene from their father. 1/2 of the males are red-eyed, inheriting a red eye gene from the mother's X chromosome and no eye color gene from their father's Y chromosome. The other 1/2 are white-eyed, inheriting the white eye gene from their mother's X chromosome and no eye color gene from their father's Y chromosome. The outcome of Morgan's Punnett square is consistent with the actual breeding outcomes. The Morgan hypothesis isn't just about predicting eye color in fruit flies. It is also relevant in explaining the inheritance of a number of genetic diseases in humans. Genes that are located on one sex chromosome but not the other are referred to as sex-linked. Nearly all sex-linked genes are found on the X chromosome and are often referred to as X-linked genes. Humans carry genes related to color blindness, hemophilia, and muscular dystrophy exclusively on the X chromosome. The fact that males have only one copy of the genes for these traits puts them at a much greater risk for these illnesses than females, as they have no chance of inheriting a second good gene that will mask the expression of a bad gene they may inherit.
Okay, so let's try one of the X-linked inheritance problems and see, for instance, if someone had color blindness or hemophilia or muscular dystrophy, what's the chance of passing that on? So on to the whiteboard, and if you would look on page 93 of your course outline, and you can see the key there that we use when we're looking at males versus females, and you can see this is different. So always remember, if you're doing a problem like this and you see that it is X-linked and it's going to be recessive, right? So recessive, that you have to link it then to the X, and you have to include the gender. So you have to put down the male and female. Let's start with the female. So the female, and we'll put down the X with the big A is going to be, since this is a recessive disorder, big A is going to be normal, and X with the little a, remember these are X-linked, only on the X, little a is going to be affected. And so when we look at the key, the female can be homozygous, homozygous dominant, heterozygous, or homozygous recessive. Now, if she's got two normal genes, she will be normal. If she's got one of each, what do we call that? She appears to be normal, but we call that a carrier. And then if she's only affected if she has a homozygous recessive condition. Okay, now if we look at the male, he's either got a big A, he has nothing on the Y, or little a. That's it. So if he has a big A, he's going to be normal. And if he has a little a, he's going to be affected. So again, he either has it or he doesn't. That's that. So our try this on page 93 is to cross a female carrier with a normal male. So we set this up as a regular monohybrid cross 2x2 box, but you have to remember to put your male and female cross. It doesn't matter which one goes where, just make sure you have both male and female. And then you link, X-link, these letters to the X. So if we are crossing a female carrier, here's the carrier. So she's got one of each. And the male is, what are we crossing here? Normal male. So he's normal. He has a big A. Okay, what's the chance of having an affected child? So if we do this, and you can see then, here's the F1, that now here's the problem with these questions. If it says just simply, what's the chance of producing an affected child? Well, out of all these kids, this is the only one affected, it would be 25%. But if it said, what's the chance of the boys to be affected? Since you have a 50-50 chance of having boys versus girls, these are the males, it would be 50%. So of the boys, 50% would be affected, 50% normal. And of the girls, 50% normal, 50% carriers. So where did this boy get this faulty allele? He got it from mom. And that's what we see often happens. These parents don't realize that, mom's a carrier, dad's perfectly normal, and all of a sudden they have an affected son who is either has hemophilia or muscular dystrophy, or who knows. So if we look at the characteristics then of, on page 93, and it says there, the characteristics. Hold on, my mic is having issues. 1 second. Okay. So if we look at, it says more males than females are affected because, again, males only need that one faulty allele for them to get it. Now, this is looking at some different scenarios here where big N is normal, little n is affected. Here's an affected male, here's a perfectly normal woman, and then in the lighter boxes, these are carriers. And so the white boxes are normal and the red are affected. But in order for a female to have the characteristic, her father must also have it, and her mother must have it or be a carrier. In other words, she has to get a faulty allele from both parents. The next one, the characteristic often skips a generation from grandfather to grandson. That's what we see here. So here's a male who's affected, and notice if he has children with a normal female, then all the girls are going to be carriers. And so the next generation, the girls are carriers, it appears to be normal, but notice then she can pass her faulty allele on to her boys. So then the next generation has an affected child. So it skips from grandfather to grandson as it's hidden in the carrier mother. Okay, so that's it for our Punnett squares. And also in this course outline, there are some practice problems for doing X-linked inheritance over on page 96, so be sure and practice those. And we're going to continue on looking at other factors that affect chromosomes and genetic inheritance of disorders. And it talks here about multifactorial disorders. Many diseases such as heart disease, diabetes, alcoholism, mental illness, and cancer have both genetic and environmental components. And so we call these multifactorial diseases. There are so many factors that come into play. And whether just because you inherit a gene for something, let's say you inherited a gene for cancer, but say you don't smoke, that's a trigger. That is an environmental factor that will trigger those cancer genes in the lungs. So you have a less chance of that happening. So just because you might have the genetics doesn't mean it's going to be expressed. And so now we have a new branch, really, called epigenetics that talks about how external factors influence your genes. So your genes are the instructions, but if they're not activated, nothing's going to happen. So here's a clip on some interesting inheritance and epigenetics.
Are you still using Google? I have something to show you.
The sweet smell of fruit doesn't normally send rats running, but when researchers paired the orange cherry almondy scent of the chemical acetophenone with a painful electric shock, lab rats quickly learned to fear it. Along the way, extra neurons sprouted in their noses and in the smell processing center of their brains, making them super sensitive to the scent. This result isn't shocking. What is surprising is that the rats' pups and their pups' pups were also startled by the smell of acetophenone and had the same extra neurons as their fathers, despite never having been introduced to either their dads or the fruity scent before. But how could the pups have inherited something that their fathers learned? Basic genetics tells us that only DNA gets passed along to offspring. Characteristics like memories, scars, or giant muscles can't get passed on since acquiring them doesn't alter the genetic code. But it turns out that instilling fear in the rats did trigger genetic changes, not in the DNA sequence itself, but instead in how that code was read and used in the rats' bodies. In every cell, biological machinery constantly translates DNA into the proteins needed to carry out vital processes. Chemical switches attached to the DNA turn genes on or off or up and down, telling the machinery which proteins to produce and in what quantities. These switches, called epigenetic tags, are why a kidney cell looks and acts differently than a skin or nerve cell, even though all three cells have identical DNA. But the switches in any one cell aren't set in stone. Teaching those rats to fear the fruity smell switched one of their smell-sensing genes into overdrive. Researchers don't know all the places in the rats' bodies where this switch got flipped, but they know it happened in one key set of cells, the rats' sperm cells. which would one day pass along the tweaked genetic material, making the next generation of rats super sensitive to acetophenone. Rodents aren't the only creatures demonstrating this weird type of inheritance. In Överkalix, Sweden, boys who suffered through tough winter famines went on to have super healthy sons, with extremely low rates of heart disease and diabetes. And their sons had the same excellent health, living an unbelievable 32 years longer on average than the grandsons of boys who hadn't gone hungry. To be clear, this does not mean that we should start starving our kids for the benefit of future generations. Scientists don't even know yet exactly which switches the Swedish famines flipped. While we have been able to connect specific epigenetic changes to health effects in mice, we're a long way off from being able to make those connections in humans. That may sound like a bummer, but it's mostly because we humans don't live in the well-controlled environment of a laboratory. And for that, we should be grateful.
Seem to have lost my PowerPoint. There we go. It's there. Okay, so again, kind of interesting stuff, but looking at these chemical switches is getting a lot of attention right now, and trying to understand what turns these genes on and what turns them off, and especially when it comes to cancer treatments and other things like that that have somewhat of a genetic component. Okay, so looking at some other situations that involve changes in chromosomes, there was a woman by the name of Marie Barr who discovered what is known as a Barr body. And what she found out is, as she started looking at cells and she started noticing that there are these condensed cells, these inactive cells, that in a pair of X chromosomes that only one X is turned on and one is inactive, and it's kind of sort of bound up into a little ball, and she called that a Barr body. And so this is an inactive X. Because a male only needs one X, a female only needs one X, so she's got an extra one. So it's a matter of chance which one of those X's is turned on and which one is turned off. And as a result of that, she can have a mosaic pattern, depending, especially if there's a faulty X involved, that can affect all different kinds of things, from coloration in calico cats to sweat glands in humans. And so when we look at X inactivation in calico cats, so a calico cat is a black and orange cat, as you see here, and there is so it's the interesting thing about calico cats in coat color, say that 5 times fast, is that coat color is co-dominant. So remember the ABO situation where A and B are both dominant over O? It turns out orange and black fur are both dominant over white. So they're co-dominant, and they are X-linked. And so they're on the X chromosome. And since one of these X's is going to be turned off, whichever X is inactive, the active X will give the black fur. And in this case, the orange X is active, so that gives the orange fur. So again, these are somatic cells, so we get this mosaic pattern. We talked about mosaics in eye color before. But so a calico cat then has to have what in order to have both colors show up? It's got to have an X for black and an X for orange, which means calico cats have 2 X's, meaning they are what, male or female? Yes, all calico cats, normal calico cats, are female. But there is a situation where you can have a male calico cat, but he's got a mutation. I'll get to that in just a minute. But anyway, so there's coat color in calico cats. We also have a situation called anhydrotic ectodermal dysplasia, which is the result of a recessive mutant gene on one X, and that results in patches of skin with no sweat glands. So again, it's X-linked. You're going to see it in females because she's got 2 X's. Anywhere that the good X is turned on, she can sweat, and anywhere there's the bad X or the faulty X is turned on, she can't sweat. So these women have to be very careful about exercising and getting overheated and that sort of thing. Now, concept 15.3 talks about gene linkage. And the question becomes, if some of these genes are so bad and they cause diseases and problems, and the theory of natural selection tells us basically survival of the fittest, those that have the most fit genes will pass them on, and you'll see them in the population. What is keeping these faulty genes in a population? And one explanation has to do with gene linkage. And that is the tendency for genes to be inherited together because they are located near each other on the same chromosome. So each chromosome has hundreds of, or thousands of genes, except the Y, as we mentioned. Genes located on the same chromosome that tend to be inherited together are called linked genes. And the key here is the chance of inheriting linked genes is directly proportional to the distance between them. So in other words, if gene A is a faulty gene, but it's closely linked to B and C are necessary for you to survive, maybe, metabolize your food or something very important, then you're going to get gene A if you get B and C. These are all recessive. But what can change this up is if somehow, remember crossing over where we exchange segments? What happens if two sister chromatids exchange segments and get that faulty A off of this chromosome? That is one thing that can disrupt linkage, but it's very hard to do. And it also explains why some traits are carried together. For example, why red-haired people usually have freckles. Those are closely linked. So as you see here, crossing over of homologous chromosomes breaks the physical connection between genes on the same chromosome. But going back to this slide, what's the chance of separating A from B compared to A from C? There's a lot more room. So it has to do with that distance between them. So is gene A more closely linked to gene B or gene C? It would be more closely linked to B, right? Because it's closer there, the distance, there's less distance between them. But if you do remember, as far as crossing over goes, remember non-sister chromatids may or may not exchange segments, but you can see this is one way to get rid of, say, this faulty A by crossing over. And that would put it onto a different chromosome, and maybe eventually it would disappear from the gene pool. That's one way you could get rid of it. But again, a lot of these are very closely linked to important genes, and that's why they keep showing up in the population. Looking at concept 15.4, we've talked about changes or how we inherit these disorders, but now we want to look at mutations and what changes the chromosome itself and causes these problems. So looking at either the chromosome number can change or the actual structure of an individual chromosome can change, and that can cause these disorders that we're talking about. So there's large-scale chromosomal alterations in humans and other mammals that often leads to spontaneous abortions. That's usually the reason for miscarriages. There's some kind of large-scale problem that occurred, and it's just a matter of chance. It just happens. It's nobody's fault, unfortunately. The results are devastating, of course, for people looking forward to that, but it just does happen. It's just a fact of life, unfortunately. Plants can tolerate these genetic changes much better than animals because plants can, even if their chromosome numbers get changed, they can always reproduce asexually through cuttings and mitosis. We don't have that option. So if our gametes get messed up, our chromosome numbers get messed up, the result is sterility. And sometimes, as I mentioned there, depending on where it's happening and when it's happening, if it happens during fetal development, it can result in a miscarriage. So when we look at abnormal chromosome number, what changes our chromosome number? How do we, or why is it that some people have, instead of 46 chromosomes, they only have 45, or they might have 47? What happened there? And usually that's a result of something known as non-disjunction. That word refers to the fact that pairs of chromosomes, and sometimes the chromatids, fail to separate during meiosis. And the result is that the gametes don't have the correct number of chromosomes. So you might have one gamete that has an extra chromosome and the other one is missing one. And that would result overall, after fertilization, in an individual having 46 chromosomes or 47 or 45. And if you have an odd number of chromosomes, what's going to happen when you reach puberty and those cells try to divide by meiosis? You can't divide an odd number of chromosomes evenly, so that causes lots of problems. So here's what would happen. So looking at meiosis 1, remember in meiosis, I hope you still remember meiosis, where we have our homologous pairs in meiosis 1 will line up in the equator, and then the pairs are supposed to separate, but this pair does not separate. And if we continue on through the rest of meiosis into meiosis 2, where the chromatids separate, and then they separate out into gametes, notice if non-disjunction happens here, half the chromosomes, or half the gametes, will have an extra chromosome. We call that n+1. So instead of, in our case, 23, it would be 24, and half are missing. So all the gametes are faulty here. And in the case of non-disjunction in meiosis 2, where the chromatids fail to separate, then you see we only have half the gametes are faulty. So in the case where they have an extra one, this is what we call trisomic. They have 3 in a pair instead of 2. Tri is 3. And here we're missing one. This is called monosomic or monosomy. And that would be 1 instead of 2 in a pair. So we have this word aneuploidy, which has to do with the results of fertilization producing either gametes with an extra chromosome, which again we would call that trisomic, or missing one, which we would call monosomic. So aneuploidy is the overall category, meaning that the chromosome number is either has an extra or missing one. Specifically, monosomic is missing one, trisomic has an extra one. And here is a karyotype. So when you do your karyotype activity in your lab, your online lab, you will see as you look through these chromosomes, you will look around and see if you see any abnormalities. And you can see also this is something you have to do on the lab practical. If I give you a karyotype, be able to tell me if it's normal or abnormal and what's wrong with it if it's abnormal. Here you can see pair number 21 has an extra chromosome. So this is what we call trisomy 21. This is a female. Here's her 2 X's. And even if I didn't see the picture of this cute little girl, I would know it's a female because of the 2 X's. And I know that because they're the same size and same shape. If they are non-homologous, not the same size and shape, it would be a male. So here you see a trisomic condition, trisomy 21. This is a pretty common type of Down syndrome. And so it is caused by non-disjunction, failure of chromosomes to separate during meiosis. But you'll notice here that they did a correlation between maternal age and Down syndrome. And notice the incidence of Down syndrome increases exponentially after age, say, 35 or so. And that is the reason why women who are pregnant at 35 or over usually have an amniocentesis or chorionic villi sampling is recommended, because the incidence of non-disjunction increases. We don't know why, it just does. And so that's why they usually want to do that. Now, if you do get a chorionic villi sampling and you get your karyotype, it's going to tell you for sure, without a doubt, the gender of the baby. So a lot of parents don't even want to know that. Some parents don't even want to get this test. But the docs that are going to deliver usually want this done just so they know if you're at a high risk and to be prepared. So it's important to have that done, even if you don't want to know whether your baby has Downs or not Downs or what the gender. If you don't want to know anything, the doc wants to know, so you have to have that done. So non-disjunction also occurs in other chromosome pairs, as it does often in the sex chromosomes. And again, it can be the failure of these chromosomes to separate during meiosis, non-disjunction. This is showing what happens if non-disjunction occurs in the eggs, where the X's fail to separate. So one egg has double X's and the other has no X. And if it's fertilized by a normal X-carrying sperm, you end up with an XXX, or what we call triple X syndrome, also known as metafemale or super female. Not that she has any extra feminizing characteristics, and in fact, since she only needs the one X, this really doesn't have a lot of phenotypic problems. We can also have that egg that has no X fertilized by a normal X-carrying sperm, and now you have a person who is missing a chromosome. This is known as Turner syndrome. By the way, these are over on page 95 of the course outline, and giving you some of the characteristics of those. So females, this is a female because she has an X. She has to have a TDF gene to be a male. But in this case, they are sterile, and that's the only viable case of monosomy to be known in humans. Any other case, like this case, where the Y fertilizes that eggs, it's non-viable, which means there won't be any viable pregnancy. Now, if that Y sperm carries a double X or fertilizes a double X egg, you end up with an XXY male. This is Klinefelter syndrome. And in Klinefelter syndrome, the result of that extra chromosome in a male has the principal effects of low testosterone production and sterility. And a variety of other physical and behavioral differences are common as well. So there's that one. And then there's one more on your list, which I don't have on this slide, which is the XYY condition. So that would be when the Y chromosomes fail to separate, the chromatids fail to separate. So you have a double Y carrying sperm fertilizes a normal X carrying egg. And the extra Y chromosome in these males does not affect fertility, but they are taller than average and have slowed mental function, and they are also trisomic. So they actually did a study to see if having an extra Y made men more aggressive, because the Y chromosome is associated with testosterone production. And they did this study in a prison, and it turned out that the prison population had a significantly it had a higher incident of the Y chromosome than the normal population, which was significant. But they couldn't really conclude that that Y chromosome made them more aggressive, or was it the fact that they had slowed mental functioning, or perhaps maybe the combination, who knows. True story. So we also have a case of something known as polyploidy. So aneuploidy is just looking at one chromosome. You either have an extra one or missing one. Polyploidy, poly meaning many, is a condition where you have increases in the entire set of chromosomes. So instead of having a haploid set, right, half the chromosomes, or a diploid set die for twice, we might have a triploid situation, 3 times the set, or tetraploid, 4 times the set of chromosomes. Once again, this is common in plants, but it doesn't work in animals. And what happens in plants is that, and you can see how this works here, essentially what happens is there's no cytokinesis. So when the cells are supposed to divide during meiosis, they fail. The cytoplasm fails to separate, and all the chromosomes end up in one zygote, as you see here. If the gametes end up instead of, so the original number was 6, so the haploid gamete should have had 3, but instead they have 6 after fertilization. Now you have 6 and 6, which is 12. That's a tetraploid situation. So we've seen this very common in plants because they tend to self-fertilize, and if it happens in the male sperm and the female eggs, they can just produce entirely new species this way. Because remember, the way we define a species is based on chromosome number. We just changed this parent species from a chromosome number of 6 to a chromosome number of 12. That's a new species. So for people who don't believe in speciation, there's your proof right there. Happens all the time. When I was in grad school, we studied a lot of plant polyploidy, counted a lot of chromosomes, so it's very common. Now, we can also see differences in individual chromosomes. So we've been talking about changes by adding a chromosome or missing a chromosome, or in the entire set of chromosomes. But what about the individual chromosomes themselves? We can have several different situations showing up, these 4 situations right here. We can have a deletion, which removes a piece of chromosomes. We can have a duplication, which repeats a segment. an inversion which flip-flops them, and a translocation where a gene moves from one chromatid to a completely different segment. So, or one chromosome, that is, not chromatid. So we'll see some examples of that. So here's your normal chromosome with genes A, B, C, D, E, F, G, H. And a deletion would be a case where, and I'll talk more about this when we get to the DNA chapter, but there are enzymes that go through and read and clip out mistakes in DNA. So sometimes these enzymes might clip a piece out, and then they might just leave it out, and that would be called a deletion. So if this happens, you might, depends on what the gene is. It could be a really important gene and cause a lot of problems. It could be some of our non-coding DNA, and then you wouldn't notice it at all. In a duplication, you can have a gene segment cut out and then put into another chromosome, that the chromatid that is, and now you have repeating segments. So that would be a duplication. This is usually not as much of a problem because you already have the original genes anyway. It just depends on where this gets put in. And sometimes genes have to be read in sequence, and if this interrupts a sequence, that can be a problem. And then you can have an inversion where a piece is cut out and flip-flop. So we go from A, B, C, D, E to A, D, C, B, E. So it flip-flopped it. Again, this is a problem, especially if it had to be read in a certain sequence. And then a translocation is a case where you take a piece out and moves that segment from one chromosome to a non-homologous chromosome, so to a completely different chromosome. And a good example of a translocation is when the TDF gene on the Y chromosome gets translocated over to an X. So let's say you inherit that X chromosome with a TDF gene. Are you a male or a female? Remember, TDF, testis determining factor, makes you a male, but the X chromosome codes for the female gender. So that is what often happens in hermaphrodites, where they produce both male and female genitalia, although usually neither one is functioning properly, so they tend to be sterile. And here's a clip on DNA mutations.
This is Tyler. Tyler, with Grammarly's help, is writing an email to his boss, Anita.
The errors or mutations that occur in the replication of DNA can have significant impacts on individuals and on species. Mutations that occur within body cells can, for example, cause illnesses such as cancer if the mutation programs the cell and all its daughter cells to grow and divide uncontrollably. Such mutations are often caused in humans by radiation and chemicals, such as those found in cigarette smoke, that break strands of DNA. As we will see, the resulting repair work often leaves the DNA with an error. Mutations that occur in sex cells don't affect the individual in which they occur, but rather, if they are carried in a sex cell that unites with another sex cell, they are carried into the next generation. Such mutations have the potential not only to affect the individual that initially inherit them, but in the long term, the species as a whole. That is because an individual who inherits a mutation has a copy of that mutation in every cell of their body, including the ones they will eventually use in the production of sex cells. If the mutation increases the fitness, as judged by natural selection, of the individual and succeeding generations, the mutation will likely become more and more common in the species population. Each and every one of us are largely the result of mutations that, starting with the first piece of DNA in the first living organism, have been successful in increasing the fitness of individuals and species as judged by natural selection. We are, therefore, the products of successful mutations and the adaptations they led to. But history is full of even larger numbers of mutations that were judged by natural selection as unfit for survival. There are three basic types of mutations: point mutations, insertions, and deletions. Occasionally, during replication, in spite of all the proofreading and corrections, a mismatched base pair in one of the chromosomes, as for instance an adenine base pairing with a cytosine base, goes unnoticed, and the cell proceeds with either mitosis or meiosis. Repair enzymes in the daughter cell that received the mismatched pair notice the mismatch and proceed to correct it. However, these enzymes no longer have any way of knowing which strand is an original parental strand or a newly replicated daughter strand. If the enzymes correct the mismatch by replacing the base on the daughter strand, the original base pair will be restored. However, if the enzymes correct the mismatch by replacing the base on the original strand, a base pair different from the original DNA will result, and a point mutation will have occurred. Insertions and deletions occur not due to any errors in replication, but rather from a break or breaks in one of the strands. Often outside sources, such as radiation or chemicals, cause these breaks. Insertions often occur when one strand breaks in a single location and one or both of the freed ends loop out. Repair enzymes fill in the resulting gap with new nucleotides, making the strand that had the break longer than the strand that remained intact. As a result of how the process of replication works, after cellular division, one daughter cell will receive a new longer segment of DNA due to the insertion mutation, while the other will receive an unchanged, mutation-free segment of DNA. Deletion mutations occur if a strand of DNA breaks in two places. The section between the breaks is forced out, and the repair enzymes draw the two remaining strands together without filling in the missing nucleotides. Again, a loop occurs in one of the strands, but in this case, it is the intact strand. Now when cellular division occurs, one daughter will receive a shorter segment of DNA due to the deletion mutation, while the other strand remains unchanged. Having seen how DNA replicates itself and how mutations occur, we now look at how the genetic information contained in an individual's DNA is converted to proteins that make living organisms what they are.
What happened again? I'm losing. Oh, there we go. Okay, so we will actually talk about that last section. Protein synthesis is where we're going next, and we'll talk about DNA and how that DNA is used to make proteins. That would be in our next section. But anyway, there is a try this in your course outline on page 95. And so if you look at these normal gene sequence, let's say it's A, B, C, D, E, F, and it gets changed from A, B, C, D, E, F to A, B, D, C, E, F, what would that be? So we flip-flop these, so that would be an inversion, right? And if we go from A, B, C, D, E, F to A, B, C, D, E, missing the F, that would be a deletion. And here we go A, B, C, B, C, D, E, F. That would be a duplication. And finally, notice this one, A, B, C, D, E, F. And then we have this X, Y, Z from a totally different chromosome. That would be a translocation. All right, and that pretty much wraps up chapter 15. Just a reminder that you should be working your genetic practice problems and also do your lab 10, 11, and 12 online. And do all of that before you take your next exam. And that exam will be ready on April 8th and open until the 30th. So that's exam 4 over chapters 12 through 15. And that wraps up this information. Now the next video I'm going to do will actually go through the practice problems. So I will go through those with you, but don't watch that video until you practice on your own so you can make sure you know where you need help with any of that. And again, I'll be passing out some live chat times that you can chat with me about any of this, or you can always email me anytime you like. So have a great day, and I will see you on the next video.